Hi guys, today we're going to be talking about Ouchterlony. Ouchterlony is the immunological technique used to detect, identify, and quantify antibodies and antigens. Basically, it's telling you like what antibodies know how to kill what antigens, which are like diseases. So the first step is to get an agar plate. It smells really bad, and you mix it with sort of like a gel. And then what you need to do is make wells. The first well that you'll have would be in the middle, which contains antibodies and human cells down here in the middle.
Next, you'll have to make 4 more wells, and they will contain your antigens. You want to put similar antigens across from each other diagonally. So let's label these A, meaning that the 2 A antigens are similar, and then B, which are also similar.
And after you do that, what's going to happen is the antigens on the outside and the antibodies on the inside are going to start diffusing and clashing into each other. So there's 3 different possibilities for this. One would be non-identity, which means that none of these antigens identify with the antibodies and no lines appear. That means that you don't have the antibodies to fight this disease. The other type would be identification, where these little gray lines, or on your agar they probably appear sort of white, all appear lining up. Get rid of all this stuff to make a square. What's happening here is the diffusion from the human cells are going out, and the diffusion from the antigens are going out, and they're combining along here, where the antibodies are identifying and say, hey, I know what that is, and they're killing and create a line of precipitation along this side. Now, if there's solid lines all the way around making a perfect square, that means, yay, your antibodies completely recognize these antigens. However, there's another possibility.
So you could end up having a partial recognition or partial identification, meaning that there would be some excess part of the line hanging off on either side. And this excess means that some of the antigens were recognized and some were not.
Which means they're kind of like diffusing out, and still they're having, again, that precipitation, but it's not as exact. So in this situation, we would have partial identification of the A and full identification of the B because their lines are solid. So non-identification occurs when all of these 4 lines all have the crossing and have the overlap, which means that none of your antibodies recognize the antigens on the outside and you don't have the resistance to them.